Bismillahirrahmanirrahim. Assalamu alaikum students. Today's lecture number 33 that is from the chapter number 4 cells and tissues. And in cells and tissue we will study the subcellular structures that are known as cell organelles. So the cell organelles, the definition of organelles is that these are the small structures that is present within the cells and perform the dedicated functions. And there are about a dozen types of organelles that is commonly found in eukaryotic cells. For example, nucleus, ribosomes, endoplasmic reticulum, plastids, etc. First organelle is the nucleus. Nucleus that is present inside the cell that contains the genetic material. Now it contains different types of components, different structures. First one is the nuclear envelope. Nuclear envelope is the membrane that is double bounded membrane that is present outside the nucleus. Okay, that is known as nuclear envelope. And next is that inside the nuclear envelope there is a granular fluid, and that granular fluid is known as nucleoplasm. Next is the nucleolus. Now there is a structure that is known as nucleolus, and that is the dark spot, and it is the site where ribosomal RNA are formed and then assembled as a ribosomes. And then there are some chromosomes and chromatin, that is the genetic material. So the chromosomes are visible only during the cell division while during interphase, while the chromosomes in the fine thread like structure is known as chromatin. This is the structure of nucleus. Firstly, there is a nuclear envelope that is dark pinkish in color, and inside the nuclear envelope there is a granular fluid that is light blue in color, that is nucleoplasm. And inside the nucleoplasm there is a nucleolus, and that is the dark spot. Next is the ribosomes. Ribosomes is the organelle that is granular structure, and that is either freely floating in cytoplasm or are bounded to endoplasmic reticulum. And now the composition of the ribosome is that it is made up of almost equal amount of protein and ribosomal RNA, and these are not bounded by any membrane and so are also found in prokaryotes. Function is that it is the site of protein synthesis, and protein synthesis is very important to cells, and so large number of ribosomes are found throughout the cell. And now the ribosomes have two subunits, larger and smaller subunits. Now when the ribosome is not working, it disassembles into two smaller units. As you can see in diagram, there is a larger and smaller subunit. When they are not working, they disassemble from each other. Next is the mitochondria. Mitochondria, that is the double membrane bound structure, and it is present only in eukaryotes. Why it is present only in eukaryotes? Because it is a double membrane bound structure, and double membrane bound structures are present only in eukaryotes. And the function of mitochondria is aerobic respiration and are the major energy production centers. Aerobic respiration that we have studied in the chapter number 7. Okay, so the aerobic respiration takes place in mitochondria, and as a result of aerobic respiration energy is produced, that is ATP. Next is the structure of mitochondria. There are two membranes. Okay, we have studied these. It is a double membrane bond structure. So the outer membrane is smooth, while the inner membrane forms many enfoldings that is known as cristae. And this serves to increase the surface area of inner membrane on which membrane bond reactions can take place. And now the mitochondria have their own DNA and ribosome. So this is the organelle that have its own DNA and the ribosomes. This is the diagram, outer membrane and inner membrane. Outer membrane is dark brown in color, while the inner membrane is light skin in color. And inside the inner membrane there is a enfoldings, that is cristae, and inside the cristae there is a matrix. Next is the plastids. Now the plastids are also a membrane bound organelle that is present only in the plants and photosynthetic protists, कि उनके अंदर ये प्रेजेंट है जो कि फोटोसिंथेसिस कर रहे हैं. ठीक है, तो ये सिर्फ और सिर्फ प्लांट्स के अंदर और उन प्रोटिस्ट के अंदर प्रेजेंट है जो फोटोसिंथेटिक हैं. और प्रोटिस्ट फोटोसिंथेटिक कौन सी हैं? वो है सिर्फ और सिर्फ एल्गी. एंड नाउ द प्लास्टिड इज ऑफ थ्री टाइप्स: क्लोरोप्लास्ट, क्रोमोप्लास्ट एंड ल्यूकोप्लास्ट. Chloroplast. Firstly, it is the double membrane, and the outer membrane is smooth, and the inner membrane give rise to sacs that is known as thylakoid, and the sac of thylakoid is known as granum. Next is the chromoplast that contains the pigment associated with bright lights and are present in the cells of flower petals and the fruits. And their function is to give colors to these parts and help in pollination and dispersal of the fruits. कि क्रोमोप्लास्ट जो है वो जो हमारे प्लांट्स हैं, ठीक है, प्लांट्स के जितने भी पोर्शन कलरफुल पोर्शंस होती हैं उनको कलर दे रहा है, ठीक है, फ्रूट्स पेटल्स जो फूल की पत्तियां होती हैं. Next is leucoplast. It is a colorless and it stores starch, proteins, and lipids. They are present in the cells of those parts where food is stored. This is the structure of chloroplast. You can see the thylakoid, and then thylakoid stacked over each other, and they have formed the grana. Next is endoplasmic reticulum. This is a network of interconnected channels that extend from cell membrane to nuclear envelope. कि जो आउटर स्ट्रक्चर है सेल मेम्ब्रेन का वहाँ से एक्सटेंड होके ये न्यूक्लियर एनवेलप तक एक्सटेंड होता है. मीन्स कि ये कनेक्शन है सेल मेम्ब्रेन से न्यूक्लियर एनवेलप तक. And now it is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough is so named because of its rough appearance and due to number of ribosomes that are attached to it. And smooth endoplasmic reticulum lacks ribosome and it is involved in lipid metabolism and in the transport of material from one part of the cell to the other. And the rough endoplasmic reticulum, due to presence of ribosome, its function as protein synthesis. And smooth endoplasmic reticulum detoxifies the harmful chemicals that have entered into the cell. G. Firstly, inner there is a nucleus and then outer nuclear envelope. So endoplasmic reticulum has extend from the nuclear envelope to the cell membrane, and this is the rough endoplasmic reticulum that is attached with the nuclear envelope, and there are so many ribosomes, that is why it serves to for the production of the protein. And next is the smooth endoplasmic reticulum that is attached with the cell membrane. Next is the Golgi apparatus. Now Golgi apparatus was discovered by the Camillo Golgi, and these are the flattened sacs that is known as cisternae. And there are many cisternae that is stacked over each other, and the complete set is known as Golgi apparatus or Golgi complex. And now it is present in both plants and animals. What is the function of Golgi apparatus? Its function is that it modifies the molecules that comes from the rough endoplasmic reticulum and packs them into small membrane bound sacs that is known as Golgi vesicles. And now these Golgi vesicles transported to various locations in the cells or to its exterior in the form of secretions. You can see these are the vesicles that have come from the rough endoplasmic reticulum. Now from these move through these stacks, that is cisternae, and now these products are for the secretion and these are the product for the internal use. So the Golgi apparatus basically provides the transport to the vesicles. G. Today's diaries learn the organelles from the cell organelle nucleus to the Golgi apparatus. Thank you.